Good afternoon, ladies and gentlemen. Thank you for your coming. My name is Zuzana. I'm a postdoctoral researcher at the Weizmann Institute of Science, and today I would like to introduce to you our project, Phase Incremented Steady-State Proteins, how NMR can reveal the folding in the human body. I'm pretty sure that you have already heard about proteins, because I can see that you experience gym a lot. And you probably know that proteins are essential for gaining muscles, and to gain the muscles you need to get a good intake of proteins. But I will tell you that they are way more important, not just about gaining muscles, but actually they are building blocks of your life. Because, for example, proteins in your eyes, they are telling you, okay, now it's a daylight, wake up. Or now it's a sunset, so calm down and go to the bed. Proteins actually keep you alive, but only if they work. What does it mean if they work? Proteins is a large biomacromolecule, and this large biomacromolecule can change its shape, and it does. But only one of all of these possible conformations of the proteins is that one which can work in your biological processes. It's the similar as in the keys. You can see these two keys and say, okay, they are both similar, they're both the same, they are from metal, maybe they are also from the doors in the same building, but only one of them will unlock your home. Because when you take a closer look, they are different. And the same thing is here with the proteins. Can you guess the difference between these two structures, or am I just playing with you and they are the same? I can tell you here that is a difference, because this part with the red string is a bit shifted. And that's the small change which makes a big difference in protein chemistry, because only one of these three-dimensional conformations can unlock yourself and can work in your body. And the problem is that when you have a disbalance in this essential movement of proteins, then it can cause some diseases, for example, Parkinson or Alzheimer's disease. And this process, when the proteins are changing their shape, is known as a protein folding. And you can imagine taking a piece of paper and just fold it into the beautiful origami. And proteins do that, and they do that fast, they do that frequently, and they can fold and refold in multiple ways. And also, even if they are folded or refolded, they are still changing the shape. And that's why it's so difficult to observe, because we don't have enough proteins at the biologically relevant concentrations. It's just a few of the proteins. When something is only a few, it's hard to detect, hard to find. Also, this folding and unfolding, it's like fast process. And the environment really influences this process. Because when you, for example, add more water, add more protein, or add more acid, the folding pattern will be absolutely different, and you are not going to the bioactive conformation you need to find in your body. But what if I tell you that as me, any of you or your siblings, even your identical twin, has its own biometric passport, the same way every molecule has its own NMR spectrum? Because when you are at the airport and there's a passport control, they can look at your photo of your fingerprint or of your identification number, and they can identify you. That is that person. And the same way chemists can do with the NMR spectrum. NMR stands for nuclear magnetic resonance. I know it seems scary because it contains the word nuclear, but it's not. It's similar to the magnetic resonance imaging you know from hospital, or at least from Dr. House series. And this picture, this fingerprint of the molecule in magnetic field, can help us provide the new information. How many atoms do I have in my molecule? How they are connected? And most importantly, for proteins, what's the shape of the molecule? How does it look like in space? And to get this fingerprint, you need a specific machine, which actually looks like a beer tank, but works as a thermos. Because every of the molecules are formed from atoms, and atoms are formed from atomic nuclei and electron shell. And the atomic nuclei can act as a small compasses. And these small compasses can align into the strong magnetic field and produce the picture of the molecule into the magnetic field, that NMR spectrum. And the machine we are observing that is called an NMR spectrometer. And why I call it thermos? Because to achieve so strong magnetic field, we need to work with superconducting materials, superconducting magnets. And they are working only at low temperature. When I say low temperature, I mean something really cool. For example, the coolest thing in the world, liquid helium. And to reduce the evaporation of liquid helium, because it's really cool and evaporates easily, you need a thermos effect of the second tank of liquid nitrogen, which surrounded the tank with liquid helium, to keep it like colder and decrease the evaporation. So if you study proteins with this incredibly good machine, then you can see that both of these structures can fingerprint themselves as differently, because they both have the different in the shape, means both have different fingerprint. And you can find them in this protein NMR spectrum. But the problem is that there is a payoff, and everything has its limit, even NMR spectroscopy. And the biggest drawback of NMR is its relatively low sensitivity. However, it's the best tool we can have. There are some problems with that still, because NMR measurements, like acquiring the picture, it takes time. And with something takes time, you cannot study processes which are happening really fast, right? And you can say, okay, so add more compasses, add more proteins into your sample, then it will be easier to study it fast. It is, but as I mentioned, when you add more proteins, then the dynamics between them is completely different. You can describe it, but it's not relevant for the biological processes. So you need to keep these low concentrations. Also, as I said, this folding of the proteins is dependent on the environment. And this environment includes, for example, the water molecules or another proteins molecules. And it's really difficult to catch with standard NMR spectroscopy. But this is why we are here, because we decided to introduce a new way how to scan proteins and other molecules called phase incremented steady state, NMR. And this is the method you can scan things really fast. For example, when we compare taking a photo, it's like this is the same. You are just taking the picture of the molecule in the magnetic fields. It's just taking its photo. And with the ancient times, it took a long time to take a photo, right? It's like people were standing there for long, and even someone was running in front of the camera, it wasn't catch. But today, with your smartphone, you can just take your smartphone and make a sequence like ta-ta-ta-ta-ta-ta-ta-ta, and you have every blink, every mimic captured. And this is the same we are doing with NMR spectroscopy. We are writing it new ways how to run these experiments. Instead of just slow accumulating of the signal, we are going to proteins and making these ta-ta-ta-ta-ta-ta. And it changes our time scale completely and enables us to study fast processes and also to observe signals which were not so far observable in this methodology. So this is the advantage of my future research, because we need just to write a new experiments. Writing new NMR experiments is just to write a new musical score. NMR instrument is just your orchestra, and you need to tell each of this part, do this, do this, do this, and that time, and that time. And what I also like about this approach is that applicable on any NMR instrument. So it means that all of the world can just get this score and employ it to the study of the proteins to fill in the vital knowledge gap we have in there. And here, I would like to thank my advisor, Professor Lucio Friedman at Weizmann Institute of Science, at the Azarieli Foundation for covering of my postdoctoral stay, and you for your attention. I'm happy to discuss the questions.